Dorks, welcome to another week of Dorks. Today our theme is freshwater critters, and I am super excited about the talks that we have today. And in general, I'm super excited because there was a crazy storm a couple weeks ago, which is why I missed the last Dorks. And then there was another crazy storm, and there have been COVID exposures, and the world has been just sort of doesn't feel like it's been cooperating. But I am here today, and we've got great talks lined up, and it's going to be great. So for folks who are new to Dorks, we have two awesome speakers who are going to dork out about a topic that they love. They talk for 15 minutes each, and they answer questions for about 15 minutes, and then we all come together and do questions together. You are welcome to submit questions that my co-host, who I will introduce in a second, Scott, and I will ask to the audience. So if you want to submit a question, there's a little Q&A button down at the bottom. You can click that and submit your question at any time, and that will come to Scott and I, and we'll share it with the speakers. And that's the background on how Dorks works. And so, Scott, how's it? You just got back from Belize. How was Belize?
I did. Yeah, it was really amazing. I love Belize. I actually teach a class there in the summer for Rice University students. So it's one of my favorite places. And this was a different trip. I was going with a group from Rice, but it was a group of alumni. And so we got to go to some places I had never been in Belize, in the rainforest, seeing monkeys and birds and lots of cool animals there. And then out on some of the islands, Belize has this amazing barrier reef system, and we spent a lot of time snorkeling out on the reef and saw some great corals and beautiful fish, some sharks. I got stung by a jellyfish, but that was okay, and it was just fantastic. So yeah, good experience.
Nice, that's great. Do you know what kind of jellyfish? Because so getting stung by a jellyfish can range from not so great to like really bad day, and so you got kind of lucky. Or does Belize not have really bad day jellyfish?
Yeah, there's some jellyfish that you wouldn't want to tangle with, but I don't think this was one of the really bad kinds, like, Portuguese man-o'-war, which isn't technically a jellyfish, but a floating thing that can sting you. And I don't know for sure what it was because I didn't see it. I did find a tentacle on me after feeling a burning sensation. And luckily I was wearing kind of like a full body, not a wetsuit, but just I was covered. And so I think that prevented me from having a much worse experience. So it really wasn't that bad. Just stung for a few minutes and then was okay.
Good. I'm glad it wasn't worse. Sounds like a fun trip.
Yeah, it was great. Yeah. And hey, how are you? It's good to have you back.
Thanks. Yeah, no, I'm happy to be back. And I'm excited. So I work in freshwater systems, or at least I did for most of my grad school career. I'm sort of transitioning to WASPs. So I'm super excited about today. And so when we have Dorks, we try to find drinks that match the theme. And I also, if I can, find a glasses cover that matches the theme. And I found freshwater fish glasses covers, which is awesome because whenever you get themed like accessories about fish, it's almost always like marine ocean fish, the pretty ones, and it's never freshwater fish, but like you could find these in Lake Erie. So that's pretty great. There's like some lepomids and stuff. I'm super excited. Also, the drink that I'm drinking is the same drink that our buddy Ethan is drinking. We are both having what's called the white salamander, which is spiced rum, Irish cream, milk, and ice. Is that everything that's in there, Ethan, or did I forget something?
Yeah, I think so. That sounds right. I didn't have any crush.
Yeah, what do you think?
Other than that, I think it's pretty good.
Yeah,
I'm drinking it out of a Squirtle cup, but
that doesn't take anything away from it. I don't
know.
And mine has lactose-free milk. Yeah. Okay. Oh, I get it. That's right. Because yeah, freshwater creatures, turtles. I get it. I'm with you. Yeah. So what Zach wanted me to point out that mine is wimpy because it has lactose-free milk because all we had left was the kids' milk. But anyway, whatever. It's still quite tasty. And mine isn't crushed ice either. It's cute, but we're all just getting by.
Yeah. No, it's pretty good, I gotta say.
So usually I max out at one glass of beer, or sorry, one bottle of beer or whatever, one glass of wine, or like one shot of liquor. And this definitely goes way past one shot of liquor. So the questions when we get to Daniel are probably going to be a lot of fun, and I'm going to have a great time today. And
Good.
you know who's probably not having a great time is Scott, because his drink looks super gross.
This is absolutely goofy. This is it's very appropriate for our topic. It's a fish bowl, but this is just crazy. It's got Nerds candy at the bottom, which started off really colorful, but now are like totally bleached, which is kind of disturbing. And then it has Sprite. Actually, I cheated. I put in ginger ale because I like ginger ale more than Sprite. But it has, you know, your kind of clear soft drink of choice with some blue food coloring. And then it's got, I don't know if you can see it, it's got Swedish fish candies inside, embedded in as like the little bonus thing. So it's like, I mean, you get the Nerds as you suck it up through the straw, kind of like a boba tea. And I feel like I just got diabetes. Like I swear, like it's so sweet. It's like sickly sweet.
I think
So
I think I'd rather drink an actual fish bowl.
Yeah, agreed.
So Kelly, as you're getting, you know, really good questions, I might be having my sugar crash and be passed out. So apologies in advance.
All right, so we're gonna do our best to still stay high level when Daniel comes on. And so Daniel is the fourth D.O.R.K.S participant who we haven't said hi to yet today. So hi, Daniel. You're going to be the only one of us who's, you know, sort of normal by the end of the event.
Yeah. Well, I'm looking forward to the questions then.
All right. Well, then on that note, should we get started, Scott?
Let's do it while we're still coherent. All right. Well, I'm going to introduce our first speaker today, who is Ethan Kocak. So Ethan is a cartoonist, illustrator, and aquarist. Did I say that right?
Aquarist, yeah.
Aquarist.
Yeah.
It was 50-50.
It's okay.
And so, which I think means that you like keep aquariums. Is that what it means?
Yes.
Okay.
Yeah.
Very cool. And we always ask each of our guests to share a weird or wonderful fact about themselves. And Ethan's weird, wonderful fact is that he once had to get a live alligator out of someone's car. I have to ask, how did the alligator get in the car and how did you get it out?
It was in Pittsburgh, Pennsylvania, of all places. And I was working at a pet store at the time. And I had become known as the reptile guy there. And this person had bought what they believed was a spectacled caiman at a flea market and wanted me to sort of formally ID it for them. And they got it they brought it unrestrained in their car and had me come out to get it. And it was about a 3-foot alligator. And I had to explain with my book, this is an alligator. It's not a caiman. It's not going to stay 3 feet long. It's going to get like 12 feet long. And then he asked me if I wanted it. So
there are so many bad decisions in a row that person made.
Yeah.
Wow.
Very much.
That is wild. Well, we're excited for your talk today. Ethan is going to talk to us about, the title of his talk is great. It's Moistly Harmless, How to Appreciate Newts and Salamanders. So take it away, Ethan.
All right, thank you.
And while that's coming up, I'll just remind everyone to feel free to submit questions via the Q&A button, and we'll be taking a look at those. And we'll be asking as many of them as we have time for at the end of Ethan's talk.
Can everybody see it okay? Okay. So Moistly Harmless is actually going to be the title of a nonfiction graphic novel that I'm working on, and it's going to be crowdfunded and probably will be out around the end of April. So that's where the title is coming from. And this is me. My name's Ethan Kocak. I am most known for illustrating the Does It Fart book and its sequels, which was on the New York Times bestseller list. And I'm also known around Twitter for doing lots and lots of avatar drawings for scientists with them and their study subject. And I do a podcast called Squamates that is about, we review reptile and amphibian papers. So I'm mainly an illustrator, an artist, and a person who's extremely interested in herpetology. And I have been keeping newts and salamanders for roughly 30 years. So I have a lot of experience, and while it might not be academic experience, it's sort of practical experience doing that. And so here's a little bit of my art. There's me drawing an axolotl. The comic page on the left is from my ongoing comic, Black Mud Puppy, which is where my handle comes from. And there's that newt is from the book, upcoming book. And this is me at a reptile show. This is my table at a reptile show where I usually sell under the banner of the newtist. And you can see some axolotls and stuff that I produced there. So to get into talking about newts and salamanders, I just wanted to cover a little bit about taxonomy. Amphibians, or Lissamphibia, if we want to talk about just the extant living amphibians, is a class of tetrapods with 3 orders. And those orders are frogs and toads, or Anura, salamanders and newts, or Caudata, and caecilians, Gymnophiona. I think I'm pronouncing those right, maybe not. I have kept all of those orders, and there's some pictures from times that I've had those over the years. That toad was my cane toad, Brunhilda. The newts there are marbled newts, and that's a caecilian, a gaboon caecilian. So Caudata means with a tail, and some folks use Urotola instead of Caudata to mean the whole clade. But this is all of, I'm pointing at it as if you can see it, sorry. This is all of salamanders and their various branches here. And that brings me to probably the most frequent question with this is newt or salamander, and what's the difference? Well, all of these things are salamanders, but not all of them are newts. Newts are here. Salamanderdae, the true salamanders, so that would be fire salamanders and their allies, the newts. And the typical development path for salamanders is an egg, and then once the egg hatches, the first thing to develop, the larva hatches, the first thing to develop are the front legs and then the back legs. So they develop sort of the opposite of anurans, where with frogs and toads, the back legs come first. And that's actually a superficial thing, because with Caudata, they're actually developing those limbs internally. But from the outside, it looks like this. And the life cycle for newts usually kind of follows this pattern, where the larvae are aquatic. Then when they fully develop all their limbs and they lose their gills, they come on land and they live as terrestrial animals, which we often call efts. And that phase can last for months or years. And then once they become sexually mature, they then head back into the water to reproduce and start the cycle again. So most newts are aquatic for at least part of the year. It's different for different species, and I'll get into that a little bit more later. And then there are also neotenic weirdos, where they don't ever leave the water. They just, conditions are too good, and they keep the gills, and they become adults underwater, and they stay that way. And so neoteny is retaining larval characteristics into adulthood. And you can see here, the most famous one is the axolotl. Everybody kind of knows axolotls, but there are also many other neotenic salamanders where they never have an adult phase. They retain their larval characteristics their whole life. The other animals in these pictures here are a lesser siren and a Gulf Coast water dog, and I'll talk about them also in a little while. So my specialty is in caring for them and breeding them, and this is an example of a Kayseri newt from Iran. They were once very rare. They're now pretty common in captivity. But that's mostly through the efforts of people like me who are just hobbyist people keeping these in their basement. So as a disclaimer to that, I want to make it clear I don't condone capturing wild animals. A lot of things that I see on Facebook are people who say, I found this animal outside, I took it and it was cold. Well, most of them like the 50 degrees outside is perfect for them, and they're not cold, they like it that way, and you kidnapped a perfectly capable wild animal. So there are plenty of these animals already in captivity and being bred in captivity, and what I want to discuss is information about helping those. There's already plenty of them. Let's not go catching stuff out of the wild. So caring for caudates in captivity, getting started, some housing philosophies, and some common problems. So if you're getting started, you're probably going to want a fairly hardy species. You're probably going to want to make sure that you can provide the right types of temperatures. So you don't necessarily want to get into this if you live in a very, very hot climate all the time and don't have air conditioning, for example. And you want to be able to provide the right kind of habitat for them. So that would usually mean an aquarium or an enclosure like that of some kind. So here's some examples of enclosures. An enclosure could be as simple as a plastic shoebox with paper towel. And you can see there's little babies there on the right are little baby fire salamanders that were born at my house. And then on the left here is what I keep those Kayseri newts in. It's a naturalistic type of enclosure where I'm aiming to produce as close as possible what their conditions would be like in the wild. So with them, they're from these mountains in Iran and Turkey, and they're limestone mountains geologically. So I tried to seek out limestone. I actually tried to see if I could buy limestone from Iran, but that was not possible because of the way things work. But so with naturalistic enclosures, you're trying to kind of reproduce as close as possible natural conditions. And both of those things have advantages and disadvantages. When you're doing this, you want to keep in mind, the species that you're keeping, the land to water ratios, because they have different needs. In regard to that, the type of material, so hardscape materials like the rocks that I mentioned, do they have bad habits like axolotls where they tend to ingest substrate? So you have to maybe consider that. How are you going to be filtering it? What are you doing for lighting? And what, as I mentioned, what temperature concerns? And here are some tips for equipment and how to do it on the cheap. So glass aquaria are usually often very available, and many of the big chain stores will often run what they call dollar per gallon sales. So you can, if you time that right, you can often get good aquariums, or people are often trying to get rid of them. So I always check like garage sales and stuff like their Craigslist, if you're brave. And then I always use, I have an air pump. It's called a Medivac blower, or a Medi blower rather, that I hook up to the whole room to do my aeration. But just a basic air pump for any aquarium with airline tubing and an air stone is often more than enough, because I tend to rely on very heavily on biological filtration. But I also do weekly water changes to help with that. And my lighting is almost always real basic LED lighting from, shop lights from the hardware store. And play sand or stones from the hardware store is usually much cheaper than buying the same thing at the aquarium store that somebody slapped a fish tank logo And I often make my own lids too. So just a little bit about aquarium fundamentals. If you're going to get into keeping aquatic animals, it's important to know some of this stuff to get the tank prepped and ready. And that means cycling. So aquariums work based around the nitrogen cycle. So with this, I just put this together to show that, you know, your aquatic salamander here produces waste, which is ammonia. You have bacteria that you're trying to establish to convert that ammonia to nitrite, which is less toxic than the straight ammonia. And then a different set of bacteria that are converting that nitrite to nitrate, which is again much less toxic. And then at that point, you remove the nitrates by either doing water changes or growing plants. And then you can trim out the plants and move them around and stuff like that to also to remove nitrates. This is how all aquariums work. And so here's some examples of that at the bottom there. That's one of my axolotl tanks. And you can see the plants growing in there. Axolotls tend to be a little bit tough on plants because they trundle over everything and lay their eggs in them. But I also use all kinds of other stem plants. And at the bottom left is not a salamander tank. It's just a tropical fish tank that I have in my upstairs. But you can see I'm growing a monstera out of the top of it because I like to use plants as much as possible to take nitrogen out. And on the right at the bottom is also one of my grow out bins. And you can see I have, that's actually much bigger now than when I took that picture, but I have a pothos that's basically growing all over my room because it's pulling the waste out of those tanks. So then feeding caudates, I use frozen foods, live foods and dry foods. I use all those in rotation and depending on what's needed. Baby salamanders, when they first hatch, almost always exclusively eat live food and won't eat anything that doesn't move. They have to develop a sense of smell before they're able to take anything else. So usually for the first several months, I'm doing bloodworms. I don't like doing brine shrimp anymore because they're kind of a pain to raise. You have to wash out the salt water, and because they're saltwater animals, they die in fresh water, so they pollute the water much faster because they die. Whereas live blackworms that I use all the time do not die. They're freshwater creatures, and if you chop them up, they just grow into more blackworms. So they're ideal. But I spend a lot of money on them. I can't culture them fast enough to feed the entire collection that I have. So I have to spend, you know, $100 a month on having them shipped to me. So some common issues that people run into when they're getting started are incomplete cycling. They don't wait long enough for the tank to establish itself. And that's true of all fish tanks, all aquariums. Newts in particular are often prone to fungal or bacterial infections, which you can help combat by using botanical stuff. Like I use a lot of Indian almond leaves in my tank as leaf litter, but the tannins in that also help to prevent fungal problems. Stress, and often stress is related to the next thing there, flow and temperature being wrong. So particularly with axolotls, for example, axolotls are really popular and they get stressed quite a lot by the flow being too strong or the temperature being too warm. So that's where people tend to run into problems the most. So then I wanted to get into just a little bit about how to care for the different types of caudates, and I broke it into aquatic, semi-aquatic, and terrestrial. Aquatic salamanders are salamanders that spend all or the bulk of their time in the water, and they're going to be the most similar to keeping a traditional aquarium or fish tank. So I would say axolotls, there's a lesser siren there, my Amphiuma, water dogs, all sorts of adult newts. There are a lot of adult newts that don't pretty much hardly ever leave the water. So those would all be primarily aquatic setups. And so this is a typical axolotl. They are by far now the most popularly kept salamander. They are originally from Lake Xochimilco, which is in Mexico City, although they're mostly extinct there because of introduced tilapia and pollution and things like that. So there is a program going to try to salvage them, but their situation in the wild is not great. They have been in captivity, well established in captivity, both for the pet trade and for use as model organisms and lab animals. But they've been in captivity for roughly 200 years. The first shipment of axolotls out of the New World went to the botanical gardens in Paris, and they still have axolotls. They have, like I mentioned before, they have a bad habit of ingesting substrate, so you never ever want to use gravel with them. You always want to use, actually the easiest thing to keep them on is no substrate. You can just spot clean much easier that way. But play sand or rocks that would be way too big for them to swallow are okay. Pieces of slate or tile. They're famous for and studied for the purpose of regeneration. So they have amazing regenerative abilities, probably in part because of their neotenic nature. The most recent thing with axolotls that you see a lot now is they were in a limb regeneration study, and part of that study was splicing jellyfish genes into them to give them the GFP, green fluorescent protein coloration, so they could be traced. And so that sort of leaked into the public sphere, and you can now get axolotls that glow green under black light. They like their water, like most of these guys, like most salamanders. They like their temperature about 65 degrees Fahrenheit. And colder is fine. Warmer, anything above 72, and you start to run into problems. So some of my tanks I have chillers for, or you have swap ice bottles during the summer, things like that. They become popular lately in part to be their inclusion in Minecraft. So now it's a lot of kids that are interested in keeping them, which I think is great. And I should mention too that nearly all of the individuals that are in captivity, while they are saved from extinction, they have been hybridized with tiger salamanders. The whole reason we have albino axolotls at all was because of an experiment in the 1960s where they hybridized them with a albino tiger salamander. This is another species that I keep aquatically. This is Necturus molari, which is a new description. They used to be called Necturus cf. Byeri, but these are from the Panhandle area of Florida, and basically nobody keeps them in captivity. So this is a project that I've kind of been flying blind on. They hide all the time. They are larger. They're not as large as their cousin, the mud puppy, but they are, which is the same genus, but they are more warm tolerant, and they get about 12 inches long or so. So they're still pretty appreciable size, pretty good sized salamander. I have them in a 60-gallon pond these days, but they all live under this one like 1 foot slate rock together. I've had that group going, started 8 years ago, and 2 of them seem to have just hit maturity. So they're a really slowly growing species, which makes sense because there are other proteid salamanders that are like that. So most famously olms, and olms are known to like not move for 7 years and stuff like that. And one of mine I didn't see for a year once, and it was just hiding under a different log. So I don't disturb them very much. I don't go in there. I go in every couple of months these days to make sure everyone's okay and to check on them. But yeah, I just throw worms in there and they get bigger. This is a lesser siren. So similar sort of look, but much longer with no back legs. And I have to keep the water level a little bit, a few inches below so she doesn't get out. They still get almost 3 feet long. They're one of the longest salamanders in North America. Extremely active. This is the most active salamander I have. She's almost always out swimming around. They're sexually dimorphic. The males have huge masseter muscle jaw for their jaws, so they have a distinctly weird looking head, whereas the females have this, you can see her flat sort of head. No hind limbs and eats constantly. This is a two-toed amphiuma. This is the longest salamander in North America. They can get just under 4 feet. They also have to be with a tight-fitting lid, and they have ridiculous legs. They do have hind legs, unlike the sirens. And I have a picture on the right there. You can see her little tiny ridiculous back legs. Reduced eyes, like sirens, they're also native to Southeast United States. They have, apparently, I've never been bitten, but I have talked to people who have. They have one of the most crushing, nasty bites of any salamander. They can require stitches. They are, basically their jaws are designed for crushing snails and mussels and things like that. So they have very strong bite, and mine is named shrimp flippers. So then we have semi-aquatic, which is more of the bulk of your newts, where they spend equal amounts of time on land and water, or they switch seasonally. So you usually want to build something that has land and water parts. These are California newts, for example. They're aquatic for about half the year. To get them to breed, you need to get them down into the 40s. So I put mine in the garage for part of the year. They are, along with their other species in their genus, they're extremely toxic. The picture at the beginning of my talk with the California newts sitting in a teapot is an homage to this apocryphal story about some loggers in the 50s who supposedly were all found dead because one of those newts had gotten into their teapot and got boiled alive. And they drank it without knowing it. So they are extremely toxic. They're one of the most toxic vertebrates known, equal or rivaling the toxins of some of the poison dart frogs. And interestingly, with dart frogs, if you've ever raised dart frogs or you've seen them in captivity, dart frogs in captivity aren't dangerous because they get the toxins from insects that they eat. But with newts, that's not the case. They are they are toxic without any need to sequester it from anything. And in this picture, they are shown here attempting to reproduce and failing at it because that's how they are. This is a Chinese firebelly newt. They have a very short larval period where sometimes they'll mature within a couple of weeks, but then they spend almost an insane amount of time as a terrestrial eft. They grow really slowly during that time, and I've had them take as long as 5 years to go from terrestrial back to aquatic. That brilliant orange coloration, which is aposematic coloration, just like dart frogs, where it's a warning color, leave me alone, is carotenoid based. So they get it from things that they eat in the same way that flamingos get their pink from shrimp that they eat. So you have to provide them with carotenoid rich foods or they just look like pale yellow. And this is a dwarf species. There are lots of other species in the genus Cynops that are 2 or 3 times as big as them, and they look similar. But these are they used to be the newt. This was imported by the millions. So when people think back to their childhood and they had newts from the pet store, this is the one they usually think of. Several years back, there was an import ban because of concerns over chytrid fungus, which is a disease that's been harming amphibians. And so the importation of salamanders from Asia was stopped. So you can no longer bring these in by the millions. So all that's here is what was left here after that. And so I think it's kind of a good thing that you can't walk into a pet store anymore and walk out with one of these guys for $10. But it is something that comes up every time I sell them at shows or something where it's like, what do you mean you're charging, $60, $70 for these? And it's like, well, because before nobody had to, chop up live worms to get them to size for 5 years. This is a palmate newt. They are from the UK and Western Europe. They're mostly aquatic as adults, but they also go through about a year-long eft phase. And they have a really neat, the males have this cool tail filament. So a lot of newts have sort of rituals where they will flick their tails. The males will do that to like waft pheromones at the females to get them interested. And so they have a neat little filament on the end of their tail when they're in season. And I included this slide because last year, one of them turned out to be this weird gold color, and I'm hanging on to that one, and his name is Nugget. You can see him next to a normal colored palmate newt on the right. And then there are terrestrial salamanders. So this is a fire salamander here in the picture, and I'll get into them in a second. But these would be salamanders that require terraria instead of aquaria. And this is an example of how you would put a terrarium together. I often just use plastic containers. I don't like cocoa fiber, which is often sold for that because it sticks to everything. And with animals that breathe through their skin, that's not great. You want to make sure that there's a drainage layer at the bottom, so clay or stone. And my super secret recipe is for soil for that is there if you ever need it. And there's ventilation is important, and also springtails. I use springtails to eat the waste that's left over from uneaten food or animal waste. So these are my Bernadesi fire salamanders. The individual shown in the water bowl there is the only female in the US currently. And there's some of her babies at the bottom right. So the first time. And this is one of the few salamanders that actually gives live birth. And they actually have intrauterine cannibalism like sharks. So the you get a litter of them, but it's not as many as you would get for other salamanders, but they're almost fully developed, which is nuts. It's just bonkers. You can see she's very pregnant in that picture of her in the dish there. And the male is the one in my hand, and he's also still doing great. And they like to accept food from tongs, and they come out and beg like little dogs. And then here are barred tiger salamanders. So this is sort of the North American convergent version of that. They are mole salamanders. They're very closely related to axolotls, same genus. So when axolotls, this happens rarely, but sometimes axolotls will go through development and they won't be neotenic. They end up looking something like this. They live underground. They can get very large. I've seen them eat mice. They will eat almost anything. And a very cool salamander. That's maybe for a project down the road. I was looking into what it takes to breed them. They haven't been bred in the US. Europe and Japan, there have been a few keepers who have, but funny enough, nobody here seems to have been able to do it. So a couple tips and tricks. Lighting is really important for salamanders. They take a lot of cues for season via photoperiod. So I have all of my lights on smart timers so that the lights come on at dawn and go off at sunset every day. And so they get the equivalent of the right amount of light. And I also keep stuff in the fridge or in my garage, as I mentioned, to hit the right temperatures. And so sometimes if you come to my house and you open my fridge, there will be friends staring back at you. This is my patented newt proofing trick. I use packing tape around the edges of most of my aquariums for any animal that leaves the water or is prone to leaving the water. Newts can't climb at a 90-degree angle. So this keeps them in the tank. It's cheap, it looks dumb, but it works.
Ethan, this is great. It looks like you're just about wrapping up. I want to make sure we have time to get to some questions.
Sorry if I went over a little. I'm sorry.
No, go for it.
This is the end. I just wanted to also mention that, like I said at the beginning, the book will be crowdfunding soon. We were going to do Kickstarter, but then they decided to go blockchain, so we had to come up with something else, and I think we have that straightened out. So in the spring, and if you follow my Twitter there, you'll get updates on that.
Awesome. Thank you. That was really interesting. I there were some species there I had no idea about. So I thought that was really cool. And you've reminded me of, I had not thought about this in a long time, but when I was in college, I had an enclosure in my room that had a bunch of different types of amphibians and some fish. And it included a tiger salamander.
Oh,
and
yeah.
Slowly but surely, all the other things in the tank disappeared.
It's so funny that you mentioned that. I considered having a slide in here, and I didn't do it because but I considered having a slide that was about species mixing disasters, because another common pitfall with new keepers is often trying to put, a bunch of different species together because we're all used to community aquariums, and it often goes very badly. So I'll just say that my recommendation is always one species per enclosure if you can. So
yeah, I learned the hard way. It was a really neat enclosure. It had, had an aquatic area, it had like a waterfall and some fish in the water and the terrestrial area. But at the end of the day, all there was Murphy, the tiger salamander.
One fat, happy tiger
all
salamander,
alone and very well fed. Yeah.
Yep, that's how it goes. Yep.
And I felt bad.
I often get asked with the axolotls, because that's, people buy axolotls the most. I get asked, can I keep fish with them? And I usually say, yes, you can, but I wouldn't keep any fish that you're particularly attached to, because eventually that's what will happen.
Wow. So if people are interested in getting any of these, do you have recommendations on where you can go to make sure you're getting ones that are healthy and, responsibly sourced and everything? What are your recommendations for places to get them?
Absolutely. Good question. One of my top recommendations is there's a website called caudata.org, that's run by a gentleman who's really one of the only fire salamander breeding specialists out there. But he runs this website. It's got a kind of old-fashioned style internet forum, but all the guys who are kind of good guys and are vetted offer stuff through there. So that would be a good place to start if you're looking for legitimate stuff. There are other, places to buy exotic animals out there on the web for sure, but you often have to be careful. So I often recommend trying to join those communities first, so you kind of know a little bit about who's a good actor in the community.
Yeah, from what you were saying, it sounds like my tiger salamander was almost certainly acquired from the wild, because you said nobody's breeding them in North America, and I definitely did not know that.
Honestly, know how most of them are acquired, and I have done this in the past myself to buy them. They're usually from bait shops. They are harvested for fishermen. They're sold in bait shops, and then you have a couple of middlemen who buy them up from the bait shops and then sell them as pets.
Interesting. So they're bait initially. Wow. Yeah. It's not often that bait becomes pets. I guess that's one of the few
well, examples. And I used to you feel bad. You don't want to see that happen. I would have thought that practice would have gone away, but it's still happening even after concerns about chytrid and stuff like that.
Yeah. You're reminding me of another story I'll share real quick, which I was fishing in one of the tributaries of the Amazon in Brazil many years ago. And the guides they had a bucket of bait, and they would hand it to us to put on our hooks. And they were calling it minhoca, which is Portuguese for worm. And I was looking at this, and it was like sliced up something. It wasn't worms, because I could tell it had a backbone. And we realized it was caecilians.
Yeah, that's awesome.
They like to use as bait because the vertebrae gives the hook something to attach to. But then I felt really awful that we were using chopped up caecilians.
That's rough. Yeah.
Yeah.
I have kept, like you saw on the slide there, some terrestrial caecilians, and basically you just have a warm box of dirt. You never actually see them. they recently discovered a population of I'm going to get the name wrong, but it's the aquatic one, and in Florida. So they're established in Florida now.
Oh, wow. Wow.
So then within a few months of that happening, there were people on some of those animal sales websites selling, Florida locale Sicilians.
They are such cool critters, though. Amazing.
Yeah. So one of the things that like really struck me about your awesome talk was that in order to do this well, the amount of things you need to know is like incredible. You need to know the natural history of the species and everything up to like water chemistry and how the ecosystems work. And how do you learn how to keep species that are from Iran if you can't go out into their ecosystem and say like, oh, this is sort of what they're doing? Like, yeah, how did you learn all of this?
So, I mean, that's kind of what attracts me to it, is the deep diving into that stuff. I don't do naturalistic enclosures for everything because it's sort of impossible. But to try to encourage the natural, when it's an animal where you want to encourage the most natural behavior and get them to breed, that's often how you go about doing it. And how far you want to take it is up to you. But in the case of those newts, the Kaiser eyes, and I also keep another species in that genus, and I have had luck with breeding them. I researched that by using Google Images and iNaturalist to get plants information about what was growing in the area and what insects are in the area. And I tried to use like Google Images to go there and look. So like for my Japanese fire bellies, I actually have some friends who are Japanese keepers who sent me images of where their local species were spawning. So it's all kinds of things. It's being an internet sleuth, but it's also sort of networking with other people. And but yes, it's a lot, and I think that's sort of the appeal, I guess. But it's a very specific niche thing. I get that.
I to be honest, I did not expect that the answer was going to be go on iNaturalist and search what you find in the same habitats. Like I had assumed that there was like, an amphibian geek in Iran who has a blog that everybody goes to or something like that.
If there is and they're watching, please reach out, because I would love to. I have reached out to people in other countries specifically to ask questions like that, for sure. With the Iran stuff, I have a friend who is he deals in like fossils and rocks and minerals and stuff like that. So I had reached out to him to ask about getting limestone, and he said, "Well, you're never going to get anything out of Iran. It's just never going to happen. But you might be able to. That same vein reaches through Turkey and up into Hungary, so you might be able to get the same geological vein of rocks in another country." So we tried that route, but it was too hard. So I ended up just getting regular old limestone.
But this is like Law and Order, natural history sleuthing, sort of like that. Yes. Is not a good comparison, but like that's kind of fun that you're doing all of this problem solving to try to figure it out on your own.
Yes. Yeah. Well, and a lot of it is problem solving. Like I mentioned the Gulf Coast water dogs. I've had them for 8 years, and I just now think I have one male out of the group. So it took that long for them to hit maturity, and there's no paper written on that. There's no one trying to keep them because hardly anybody even knows what they are. They were just named as a species this year. So it's a lot of it is, if there is anybody else keeping them and you can compare notes, that's always helpful. But sometimes you just have to take your best guess and see what works.
I know I'm hogging the floor, so I'll let it go in a second, but like, so you've learned so much on your own. Do you have like a blog where you say like, hey, people who want to keep this newt, here's what I did and here's what I learned? Like, is there like a scientific paper that you could write about this stuff?
Maybe. I guess that's not occurred to me, but I have written up stuff before. I used to do a blog about aquarium stuff, but it kind of fell on the wayside. So I was doing some of that on my Patreon. to varying, to varying degrees. Yeah, I used to run a blog that was about how to do marine aquariums for dirt cheap without, spending any money. So one of the things that I had on my list of things to do there was to try to keep, to really challenge myself, was to try to keep a marine aquarium going with Victorian era equipment and rules. So based on, because I have the handbook from Philip Goss, who coined the word aquarium, and from 1864, I think it is. So following his book and using his rules and seeing if I could do it. I have never done that, but that's one of the things I'd like to do on Patreon or something at some point.
That's totally fun.
Victorian rules.
Daniel,
aquaria.
did you have... I like it.
Yeah, Ethan, I did have a question actually, because I was great talk, very interesting, by the way. But when you were talking about how you were trying to, at one point, like the Iranian limestone, you were trying to get that kind of, I guess, match the substrate to kind of where they're originally from and all that stuff. I wonder if you do that with vegetation at all, or do you find that just there's certain vegetations that you just use and it tends to work for everyone, and so you don't really need to bother with it at all? I was just curious.
Ideally, it would be the first situation, but in reality, it often ends up being the second situation. So I did try to find plants that might be of it. The problem is that the stuff that's really all around there is, often will be some random scrub plant that only grows there that no one cultivates or sells as a house plant or anything like that. So the chances of getting it are slim to none. So then you think, okay, well, is there anything that's functionally similar that's maybe from a similar part of the world that I could use? And so sometimes there is. I use a lot of aquarium plant staples when I have to, like Java moss and Anubias and stuff like that. But in the case of that particular one, I did find that field mallow, which was originally native to that area but is now invasive everywhere, could work. But I didn't end up using it because I found that it's also famous for like destroying concrete, and I didn't necessarily want to put that into an aquarium that was made of glass. And but yeah, I would always, it would be amazing to like go to a place, collect samples, and come back with it and use that as the basis to start. But then you also run into all these sort of legal issues about, is that, or even ethical or moral issues, is that the right thing to do? So it can be a little tricky.
Yeah, absolutely. But thank you.
Thank you.
Once we should transition to Daniel's talk in a second here, but like one thing that strikes me is that Autumn Anglin a few weeks ago, and you both started your talks with like, well, I'm not an academic, but then you went on to like say so much more about the study organisms than I think anyone in academia would know, because like you're actually interacting with them. And I feel like there's all of this amazing underappreciated knowledge and expertise. And it's so exciting to talk to people who like know all this stuff. Like anyway, I'm impressed.
I think that's a common thread with a lot of hobbyist keepers. I think you would find that if you talk to, especially I've noticed it in aquaria, there are whole species that exist at all because of dedicated hobbyists keeping them in their basement. It's a real phenomenon. It's a real thing. So absolutely.
I have to just jump in quickly just to add to that. Like, I totally agree, Kelly and Ethan. And I study ants, and recently this same thing has happened in ants, because there's been a movement of people who are keeping ants in much more interesting and functional enclosures than like the old Uncle Milton's ant farm that some of us had when we were kids.
Yeah. Is it the Ants Canada guy? Because I love him.
Yeah, that is one of them. Exactly. Yeah. And so that actually has helped some ant researchers to find new and better ways to keep ants alive in captivity for like experiments and behavioral observations and stuff. So I think that is really spot on, that we can all often work together between
dedicated weirdos in
researchers.
basements and academia. Yeah.
We need more of it.
It's very important. Yeah. So Ethan, I'm going to ask you to unshare your screen.
Yep.
And while you're doing that, I'll note that I love I know this is a bad image to be saying this on because you can't see the animal's expression, but like I love the expression on your animals' faces in the books that I have that you've illustrated.
Oh, thank you.
You are like a master of expressions.
Thank you very much. So I'm hitting stop share, correct? Yeah.
Yes, please.
Are we okay?
All right. We're good.
Okay.
And so while Daniel starts to share, I will start to introduce Daniel. So Daniel is a friend of mine, and he's going to be talking today about plastics and fish. He's an associate professor of biology at Fitchburg State University. And I am so excited to say that because Daniel and I met in the same lab when we were working on our master's degrees at Bowling Green State University. And it's so fun to be able to announce that your friend has a tenured position. Because, you know, when we're in grad school, we're all like, is it going to happen? And yes, it happened. And so Daniel's fact is that he's an avid comic book collector, and he's been collecting for 30 years. He has over 1,500 comic books dating back to the 1960s. And I will note as another fun fact, Daniel and I, when we were in grad school, our job involved quantifying the impacts of the coal-powered power plants on Lake Erie on the fish populations. And this job involved things like jumping into a dump truck full of dead fish under a noonday sun and using rakes to shovel flat this truck full of stinking fish and then taking buckets to subsample and try to figure out how many fish are in the entire dump truck, and then identifying those fish and trying to figure out if they had infections that were going to kill them anyway. And Daniel, am I remembering all of this correctly? Is that your memory of the best job in the world?
Oh yeah. I actually feel like I still have those boots, and I still feel like I actually find little scales on them from time to time as I'm cleaning stuff up or using those boots for other dirty work.
The good thing is every job I've had since that job, I compare to that job and think, not so bad.
Yeah. Oh yeah.
Do you have do you have a favorite comic book in your collection that is like your prized possession?
Oh God, it's hard to pick just one. Honestly, I mean, I've always just been enamored with the older stuff. To me is always just amazing. So the earlier stuff, just the way they did it back then was a little bit different. The pacing and the styling of the drawings and all that stuff was so much more done by hand. A lot of things now are more computer aided and that kind of stuff, which is really cool. But at the same time, I find sometimes the earlier stuff is just very, very different, and you can see the history come out in it, which I find really interesting in many respects. So I can't think of one in particular, but they're usually the older stuff. I just find a little bit more the history behind it is very, very interesting to me.
Do you like Gold Age or Silver Age?
They just don't make them like they used
to. Yeah, don't make them like they used to, and the technique was different. Silver Age was pretty much where I stopped. There's a couple of characters that I'm mostly interested in, so they don't go much beyond the 60s, which is where the Silver Age is. Or much earlier than the 60s, I should say.
All right. Well, I could actually talk about comic books all day, but we are here to talk about plastics and fish. And so I will shut my mouth and turn off my camera, and I'm looking forward to your talk.
Thank you. Thanks, everyone. Everyone can see my screen okay, I think so. Yes? Okay, great. Perfect. Just making sure. So thanks, everyone, for tuning in today. I'm really excited to be here. Really excited to be chatting with you. Thanks to Kelly and just everyone for setting this up, for being here. Thank you, Scott. And thanks for inviting me. I'm really happy to be here. So I'm going to talk today a little bit about plastics and fish. Before I begin, though, I'm going to talk about some of my own research that's going on. I will back up and talk about it in a broader context before I get into my own particular realm. But before I do that, actually, I just want to mention that throughout this talk, I might use the word I or I might use the word we. I tend to bounce back and forth, but it's not just me up here when I'm talking. It's really not just me. There's a variety of students that were involved in a lot of this work, Maeve and Nicole in particular. I have several collaborators that I'm working with and that have been huge helps along the way. And you can't get by without the funding and all the landowners and towns and cities that I work in order to get to those streams. So science is a collaborative process, and I really, and I do want to make sure that I say that, because even though it's just me here talking to you, it's really not just me here talking to you. So let's talk about plastic and fish. You may or may not have come across this. This is starting to become a more common and get a lot more common in news articles and get essentially a lot more attention in the scientific literature as well. You may have come across some of this before. For instance, you might have heard of the Great Pacific Garbage Patch that's out in, obviously, the Pacific. That's this giant floating island of all this marine debris that's out in the Pacific Ocean. Not all of it is plastic, but obviously, but a large chunk of it is. And then there have been other attention-grabbing headlines. There was a prediction a few years back that by 2050, there will be more plastic biomass, more plastic mass in the ocean than there are actually marine fish. There's been some debate as to whether that's truly accurate or not, whether that prediction is really going to pan out. But I think the prediction and the attention it gets just highlights the point that we are seeing this discussed a lot more, and we are seeing a trend overall in that there is more and more growing plastic pollution out there. More recently, I'd say within the past, 5-ish years, give or take, maybe 10, we're starting to also see a lot more attention focused on what's known as microplastics. And microplastics are, as the name suggests, just tiny pieces of plastic. They're defined as a piece of plastic that's 0.2 inches or 5 millimeters in length or less. And they come from a variety of sources. If you're not familiar with microplastics, you can find them and come from cosmetics, manufacturing and industry, clothing, also just improper waste disposal. And sometimes these microplastics are manufactured as microplastics. So cosmetics, for instance, some cosmetics have or had microbeads in them. Those are oftentimes microplastics. But other times microplastics can get into the environment from larger pieces of plastic, what they oftentimes call macroplastics. And that's just as these large plastic pieces degrade over time, they leach out microplastics. Think of, a single-use water bottle that you get from the grocery store. You put it in your recycling bin, but then you put your recycling bin on the curb or what have you, and the wind blows and it knocks a couple of things out and blows it into a nearby stream. That thing could sit there for years, and as the sun beats down on it, as the stream flows around it, as the wind goes and chips away at it, you get all these tiny little fragments of plastics coming off of that. And those are microplastics as well. And we are starting to see them show up in fish. Again, probably pretty recently, we're starting to see a lot more attention paid to the fact that ocean fish are consuming microplastics. We're finding them in a lot of the ocean fish and marine species that we eat, that you get at your dinner table, that you find, in the restaurants and in the grocery stores. And overall, a lot more research has been focused on marine fish species. You might be saying, Daniel, this is supposed to be a freshwater talk, but I'd like to point out that there is some potential reasons for why a lot of this focus has been on marine species and marine environments. First off is that from an economic and commercial standpoint, there's a lot of value in these fisheries and in these oceans and in these environments. Again, most of the foods, the shellfish and the fish that we eat come from the oceans. There was also for a long time this assumption and belief that if we wanted to stop the plastic debris from getting into the ocean, all we needed to do was just was to improve our sewage treatment plants. We know that now that this is no longer really correct, but there was this belief for a very long time that that was the way to handle things. And so how do you measure, well, if these sewage treatment plant adjustments are adequate enough? Well, you would go out and measure the oceans, because that's where most of the things ultimately feed into. So you're probably again wondering, well, what about freshwater systems? Can we talk about freshwaters? What do we know about freshwaters and freshwater fish? Unfortunately, the answer is not much. When we look at microplastics in freshwaters and freshwater fish species, a recent review was pointed out that our knowledge, while it's been growing, is still really much in its infancy compared to what we know about the marine systems. And I'm hoping that I probably don't have to tell anyone this that's listening, but should we care about this? I think so. There's some important reasons why we shouldn't just leave all of our studies to the ocean and focus on the ocean. There is some economic and recreational importance to these freshwater fish and these freshwater systems. Swimming, kayaking, freshwater fishing is a big recreational opportunities. There's also some certainly economic reasons from a commercial standpoint, but also we can't forget that most of the world's drinking water is coming from fresh water. And so anything that's going to impact the fresh waters is going to potentially impact our drinking waters. And not just human drinking waters, but the water we use to irrigate crops, the water we use for our cattle, and even the non-domesticated animals that are out there in the wild. If we're interested in conserving nature, if we're interested in preservation, we need to understand this, because it's very unlikely that what happens in the freshwater is going to stay only in the freshwater. This is not Vegas. Things are going to move up the food chain and through the ecosystem and through the food web. And so that is what has gotten me interested in trying to understand a little bit more about what's going on in freshwater systems. And that's what I wanted to talk to you a little bit about today. Some ongoing work looking at microplastics in freshwater streams and a freshwater fish species. So what we did is we went out to 14 streams here in north central Massachusetts. We went to the very small streams. Sometimes they call them kind of like the headwater streams. These are these very small ones that you can, easily wade in. They don't get extremely deep or extremely wide. Why did I do that? Well, because these are really the kind of almost like the beginning streams. They'll feed into larger streams, which then feed into rivers, which then eventually feed into the ocean. So we don't know too much about what's going on in microplastics in freshwaters in general. And these are some of the beginning freshwater systems. So what's going on with microplastics in there? So we went out and collected some water samples to look for microplastics. In 5 of the streams, we also collected fish. I collected a small freshwater fish species called the black-nosed dace. As you see in this picture on the bottom right, it's a small fish. Each one of those lines is a millimeter. So these fish only get to, an inch, maybe 2, if they're lucky. And why do we pick them? Well, they're very common around here, but they're also an important link in the freshwater systems around here, ecosystems around here. They feed on a lot of aquatic invertebrates, but then they are a food source for a lot of other organisms in those streams, larger fish, but other birds as well as other organisms as well. And the microplastics were counted under a microscope after some kind of complicated chemical digestions. And I'll be happy to explain more about the chemistry behind the methodology if you're really interested. It's a little complicated, but I'm happy to talk about it later if someone has any questions specific to that. But so what we did is we extracted these microplastics from the water and from the fish, trying to get a sense of what's going on in some of these very small streams and this very small fish species. And so what did we find? Well, if we look at the streams first, you'll find the streams on the far left-hand side of this graph going down that column. And then the number of microplastics in that water sample is across the horizontal on the top. Again, 14 streams, and in all 14, we did find microplastics in every sample. There's a bit of a range. Most of them fell, as you can see, about 100 pieces or less, maybe 50 to 100 or so. There was one that was that had a lot, but we did find them in all of our sites. And again, did see a little bit of a range, but we then collected, as I mentioned, 5 different fish, 5 fish samples from 5 of these streams. And so these are the ones that we collected fish from. And what did we see from the fish themselves? Well, in all 5 sites, we did find microplastics. Here on the bottom horizontal axis, you'll find the 5 streams. They're just in order that was on the previous graph. And then along the vertical axis, you'll see the average number of microplastics per fish that we found in those 18 to 22 fish, depending on the site. And so you will see a smaller range than we found in the water. Here, they fell from about 20-ish pieces up to about 60 or 70 pieces or so. So we did find microplastics in fish from all 5 of these sites. Small range, but we did find microplastics. And to give you a sense of what the microplastics look like, just to give you a little bit of idea, they come in all sorts of different sizes, shapes, and even colors. If you look in this middle picture, you'll see this little green dot in the middle. That is a microplastic. If you look here on the right, you'll see a couple of these threads, and there's one in particular that is kind of reddish or pinkish. That's probably a microplastic. There are some other threads in here that are more colorless. That's a little more debatable whether they are or not. I'll come back to that later on and talk about that. And it's not uncommon to find a diversity of colors and sizes and shapes of microplastics. In the literature, both in the freshwater and the marine systems, you do see that they classify them into different types. There's threads, there's beads, they do classify them by shape as well. Looking at a couple more patterns to what we've found so far, interestingly enough, the size of the fish doesn't seem to matter in terms of how many microplastics they have. If you look on this horizontal axis, you'll see the length of the fish from about 40 to 80 millimeters. And on that vertical axis is the number of microplastics in that fish. And it's relatively constant. There's that one little outlier, but it's relatively constant. The size doesn't seem to be hugely important in how many microplastics are actually found in that digestive tract. Looking and comparing what we found in the fish to the found in the water, surprisingly, the amount in the water and the amount in the fish weren't always related to each other. Going in, you would have thought that stream with a huge amount of microplastics, you would have seen a ton of more in those fish than anywhere else. And really, those fish didn't have many more microplastics than any of the other streams. And so there is obviously, again, smaller range in these fish than we do in the waters. But the amount in the water doesn't seem to have a huge impact on how much the fish are actually taking into their bodies or into their digestive tracts. So what are we seeing? Well, yes, there are microplastics even in these small freshwater streams. We found them in the water. We're also finding them in the fish. These small fish are consuming microplastics, or at least pulling them into their digestive tracts. And I always go back to, well, why didn't the size of the fish matter, or why didn't the amount in the water matter? And my best guess right now is that there may just be some upper limit to how much they can take in. There's some maximum amount of microplastics the fish can take in, and beyond that, it doesn't matter too much. Now, that would explain the water pattern that we saw to a certain degree, even to the size of the fish. Of course, the size of the fish, we're only talking about a very small range of sizes. So it's possible that if you looked at, a fish species that could get, have a much greater variation in range, we might see a different pattern here. But at least for the small species, it may just be that there's some maximum amount that they can take in. It's just a kind of per rate amount, and that's all there is to it. There's still some work to be done. As I mentioned at the very beginning, this is still a little bit of an ongoing work. I have some results, but we're still confirming a couple of things. The one thing that we really want to delve into, and we're working on this right now actually, is just using an infrared spectroscopy. This device, which is, as you can see on the left-hand side in this picture, it's pretty small and unassuming. It'll give us a spectrograph, a spectrometry reading of the absorbance of the materials. And why is that important? Well, it can confirm the presence of the microplastics in the sample. Up until now, I've been calling things microplastics, and I really should be using the word presumed microplastics because they could be synthetic, but non-plastic. It could be rubber or some other synthetic materials that's not actual plastic. Like I said, some of those threads that we saw, that I saw the red one, but then I saw some of the white ones. It's possible that those are, the red one is, but the other stringy ones that are whitish are not a plastic. But the other very valuable thing that this can guide us with and help us with is that it may identify which types of plastics are making up the microplastics. As you can see on that picture on the right, different plastics have different absorptions. And so we may be able to identify which plastics are really making up what is in the water and what the fish are consuming. And that may provide us with a lot more interesting questions and things to look at down the line. So still some work to be done, but we are finding some interesting patterns so far. And I will stop there to just say, again, thank you for listening. Thanks again for inviting me. I hope you are found this as interesting as I have. If you have any questions, I'm happy to take them now. If you have questions later on, here's my email address, and I'm always happy to chat later on as well. But yes, I'm very excited to be here, and I do appreciate everyone listening. Thank you very much.
That was a great talk. I'll be honest, you killed my white salamander buzz, but it's it's an important topic. And so, yeah. So to be honest, I'm like really surprised that the headwaters had so much microplastics. Like I would have thought that would be a good control site. And like, it's really depressing that there's so much microplastics at what I would have imagined were control sites.
Yeah, so I've had this conversation with students before too. It's like this work, every time you get like, you look under the microscope and you look at it and you see something, you're like, wow, this is interesting. And then you're like, oh, this is really sad. It's a very conflicting feeling on the inside. We need to know what's going on. So, I mean, I'm glad we're pursuing it and just trying to understand what's going on, but it is certainly it gives you a very conflicted feeling inside. It's not something like, yay, we found this. This is wonderful. And there's some other research I've done where I felt like that, and this is not one of those.
Yeah. A positive result usually is exciting, but here it's depressing. But so if I can try to play the devil's advocate. So like when my kids were young, they both went through phases where they put a ton of stuff in their mouth that they shouldn't have. And every time they swallowed something they shouldn't, I thought it's going to pass through and it's going to be fine. With microplastics, what are the bad things that happen when a fish consumes microplastics? It sounds like a stupid question. The answer should be obvious, but what are the bad things that happen when a fish consumes microplastics?
It's not a stupid question. I mean, there's actually, because this field is so new, there's still a lot of people asking those questions. How concerned should we be that we're finding, X number of microplastics in the digestive tract of the fish? How much of it is getting into the fish, into the muscles and into the meat or into the rest of the body? And how much is it just passing, in and out sort of thing? That's a good question that I actually don't think we have really good answers for. Looking at what we know in general, if you look at plastic pollution in the oceans, for sure, we see examples of fish getting, suffocating, if you will, or getting clogged up and they die because they ingest this. With the microplastics, that's probably less common and probably not as happening as much. But there's a chance for these chemicals to leach out into the fish themselves. Even if the plastics don't stay, the chemicals that make up the plastics may leach into the fish, which could, who knows what that could do. I mean, there's potential for disruptions of some of their biological functions, potentially. There's potential that, even if it doesn't harm the fish itself, well, what if it bioaccumulates in some other level of the ecosystem? If birds are eating these fish, maybe they'll affect the birds, but the fish are fine. I actually don't think we have a good answer to that, and it's partly because this is still relatively new topic.
Yeah, I mean, I remember when my kids were born, I avoided some plastics because they had bisphenol A, which was an endocrine disruptor.
Right.
It mimics estradiol. Is that right? And
Right.
And then I heard that the new plastics you get without the bisphenol A, actually that structure is close enough to estradiol that it sort of has the same endocrine disrupting effects. And it's just like, there's all this stuff that it seems like we don't even understand yet in the species we care about most.
Right.
How, probably tons to learn about what's happening in these fish. And anyway, I'm glad you're working on it.
Yeah, and we know they're in the fresh, we know if they're in these smallest fish in these smallest streams, they're probably in a lot more places that we haven't had a chance to look yet. And I think there sometimes is value in starting at these very small little, what you would think of as pristine, and get some answers there, and then you can build on it. I do feel like there's value in that sometimes.
I was also going to ask about the bioaccumulation thing. Like, do we have evidence that the microplastics that start off in these small fish living in small streams are going to end up in bigger fish or in other animals? But I'm also wondering about, like, if they don't get eaten up, are these plastics going to end up in larger bodies of water? Is that the assumption that, like, if something doesn't consume them, then they're going to probably end up in a lake or in the sea eventually?
Yes, I mean, these things float for the most part. They don't settle very well. So I think I feel much more confident in saying that it's probably they're going to drift downstream. They're going to, like you said, accumulate maybe in a larger lake or made up with other streams into larger rivers, and you get a larger accumulation down there. I feel more confident in saying that that's likely than knowing for sure that it's going to build up in the fish itself. Definitely think you're going to get these downstream effects where they could become greater and greater concentrated or more abundant anyway as you get further downstream. I think that's very, very likely because they float, they don't sink. And so I would expect that, yes.
Ethan, did you want to ask a question?
Yeah, so I noticed that some of the plastics look very thread-like. Is it possible that the fish are ingesting those because they're worm-like, or is it passively getting into them somehow?
Yeah, it's a very good question. My understanding is most of, at least again, when we know more about from the ocean than we do from the freshwaters. But my understanding is most of it seems to be passive ingestion. But there is some evidence that some fish view it, especially the smaller fish, like even the larval fish, like view it as food, or they can't distinguish it from food. And so they will actively, I don't want to say seek it out, but they will actively, if it's drifting by, they will go for it. So I think it is a mix of both. And you're absolutely right. Those strings certainly do look, worm-like or food-like. If I was a fish, I could easily see why you might actively go for that.
Yeah.
Yeah, I found myself wondering if part of the disconnect between the amount of plastic she found in the water and the amount of plastic she found in the fish had something to do with like food availability. If the site that had a lot of plastic also had a lot of other organisms that the fish could eat, maybe they didn't, try new things out and accidentally eat the plastics or something like that. But you're right, it is surprisingly level across all of the different sites, which suggests that there's some limiting factor like what you suggested, but it's weird.
It is weird, and there's certainly much more that should be done on that. But yeah, I certainly would have expected the fish with the highest amount of water microplastics in the water to have a higher amount in their digestive tract, and at least for that wasn't quite the case, but it maybe with larger fish or something like that, it could have been, or if you have more sites, maybe you'll see that. I don't know. It
sounds like an
interesting.
It does sound like an argument more for passive ingestion than right.
Right. Yeah.
And is it the case that the microplastics that you're seeing in the water are of the size that like are ingestible? I mean, it's not like you're finding larger plastics than these fish can fit in their mouths, because that, I mean, obviously that would be one reason why you wouldn't see a correlation, but if they're all tiny plastics, then they're all potentially ingestible.
Yeah. I mean, that would certainly make sense. We purposely, we filter. I don't think we purposely sieved out anything larger than the 0.2 because we were interested in the microplastics. So we never really sorted through what was in there to begin with. But you're absolutely right. I mean, the size, fish are what they call gape limited. The size of their mouth impacts how much they can ingest and what size they can ingest. So you're absolutely right that should play a role for sure.
We have a great question and a comment from Sean Lapatino, who gave a talk at the very first D.O.R.K.S. So she's a pathologist who gave a talk on the language of cells. So she said, I would love to know more about the extraction of the plastic from the fish. And she said, as a pathologist, sometimes teasing tissue out of plastic medical devices is significant. So that sounds like accumulation in humans. But anyway, Sean would like to know more about the extraction.
Yeah, it's thankfully it's gotten to the point now where it's sort of standardized, and so because I am not a chemist by any means, and so I am glad someone else figured out the chemistry behind it and how to extract these things, because I don't think I would have been able to figure that out if I had to. But it's now become it's kind of a multi-step process. What they wind up really what's important, what's part of it is that and it differs a little bit by the water versus the actual fish material, because you've got to deal with a lot more organic material in the fish digestive tract. So you tend to do a oxidation phase, which tends to help to digest some of the materials, and then you use hydrogen peroxide as well, which helps to digest a lot of the visible organic materials and gets that out. And then they use and you tend to use a nice big muffle furnace or something like that to help ash a lot of the material. And then from there, the last step tends to be a density separation, because again, these things float. And so by using a saltwater solution, you can actually get the plastics to float to the surface and a lot of the organic materials to settle out. So that way, then you can pass the liquid phase through a sieve. And mainly, as long as the sieve is the right size, anything passing onto the sieve itself should be plastic or at least synthetic in nature, and most of the organic materials should be gone from that. And if anyone wants the exact specific, step-by-step instructions on how to do that, I'm happy for them to email me, and I can send them the exact specifics. How much iron sulfate you need and how long to burn it off for and all that. But that's just the kind of a general sense of what you would do.
Sean wrote back, fascinating, thank you. And I'll note that sounds like a lot of work.
Again, I'm glad I'm not the chemist trying to figure that out, because we were able to take something else and run with it, and with a little bit of trial and error, it worked relatively quickly, which was nice.
We have another question from Jimmy Acevedo, who is asking, pointing out first that with waste plastic representing a massive reservoir of chemical energy, we're finding new microorganisms that metabolize it. And he asks, do you think evolution will produce something to decompose plastics on the human time scale?
That's an interesting, very interesting question. he's absolutely right that we are they are starting to find microbes on that great garbage patch that seem to have the ability to break down some of those materials, which actually could be really interesting. Could potentially help to deal with some of this pollution problems that we're seeing, which is I'm reminded of that quote, right? Nature finds a way sometimes, right? But it's
Jurassic Park, right?
Jurassic Park, yes. It's a very fitting quote. I didn't realize it when I first heard it as a kid, but it seems to pop up over and over again.
Jeff Goldblum is why.
Yeah, right. Now, whether so evolution, I guess, is starting to we are starting to see some things evolve that can handle that plastic. Whether it needs a very large source of plastic is kind of what I wonder, like, will we ever see these things evolve in a freshwater stream that doesn't have a giant garbage patch floating around in it? It's a good question. I don't have a good answer to. I mean, the more opportunity there is, the more likely evolution is to evolve something that can handle those challenges. So I wish I had a better answer than. It's an interesting question that we are seeing a little bit out in the ocean. I wonder if it'd be wonderful in a sense. If it has to happen relatively soon, I feel like, because we are just becoming inundated a lot of these systems with plastics, and not to sound like a naysayer, but I do get worried that we are going to run out of time before we need to do something else to deal with it, or else, or we're going to really ruin something before it's too late, sort of, is what I'm worried about always.
I think that's a valid concern, unfortunately. So one question that I ask myself a lot, so this is a question for both of you, both Ethan and Daniel, is so like sometimes when I'm squashing fish brains and counting the parasites on the brain, I wonder to myself, how did I get here? So Ethan has been interested in this for 30 years, he says, and I think he said in his talk. And then Daniel, how did you end up searching through fish guts for microplastics? So for both of you, what paths took you to where you ended up today?
Ethan, would you like to go first?
Sure. I guess a really winding one that I couldn't necessarily reconstruct quickly or succinctly, but I started out with catching stuff from the yard. I said don't take stuff out of the wild, but I did do that as a little kid, and it kind of progressed from there, and just a real interest in natural history and stuff like that, I think.
Yeah, I mean, I will echo that a little bit. I've always been, even as a kid, I've always loved animals, always been interested in nature and just the environment and all that. That's for sure, and that's what led me into science to go into, not only for undergrad, but then to go to grad school and all that. What got me interested, or led me down the microplastic path, honestly, is sort of the nature of just my job now. I'm at a small, primarily undergraduate institution where the students are our focus. We focus a lot, on being good teachers, but also we value the research as a way for students to get these hands-on kind of outside the classroom experiences, and which is really very valuable. And so I've always taken the approach of I have areas of interest. I have areas that I want to look at, but I try to keep it very broad. So I mean, my broad thing is freshwater fish, really, and even that sometimes I violate that a little bit and go into the marine systems. But I try to have students develop research projects, develop questions that they're interested in, and then I kind of help them go with it from there. So, I mean, the microplastic work was not my idea at all. I had been familiar a little with plastic pollution, but very little about microplastics at all until a student came actually to a colleague of mine and expressed interest in it. in it, and then she and I started talking more, and it just kind of snowballed from there, actually.
That's awesome. I love these stories about it was a student-driven project, and now hundreds of hours have been spent, and you've learned something fascinating, and it was all following the desires of a student. That's awesome.
Yeah, I think that's the important things. If you're not interested in what you're doing, or someone just tells you to go do this, you're not going to be as invested in it. I feel like you're not going to get as much out of the experience. You'll get something either way, but you won't get as much, in my opinion.
I feel like the key to being successful in life is being able to stick to a project long after it's made you feel like you're going to lose your mind, because you need to get over that hump to finish it. Right. And if you aren't working on something that you love, it is that much harder to get over that hump in every project that you hit. And so yeah, I think picking projects that the students love is awesome, is an important way to get them to actually go through the whole process.
The ADHD part of me just sort of screamed internally at that, but yes, you're right.
Kelly, are you saying that in part as someone who's closing in on the end of a long book project?
Oh, I almost every project that I've worked on has had a phase where I'm just like, I'm not done, but I'm done. I do not want to think about this anymore, but I have a couple more months where I've got to do it. And you've just got, and yes, the book project is that, but the fish brains, I think it was like 256 fish. Each one of them had something between 500 to 3,000 parasites on their brain, and they were all tiny. And I was trying to figure out if they were young or old parasites by looking at how dark their eye spots were. And at the end of every day, like, my hands hurt, and it was exhausting. But I cared about the question. So every day I was like, another day of this, because I need to know what those parasites are doing to the fish. Mm-hmm. And it wasn't much, actually. It turned out I was wrong, but it was an answer.
Well, speaking of persisting, I was going to say, speaking of persisting, through something beyond the point at which it's enjoyable. Look, I did it.
Wow.
And
I mean, yeah, me too.
I have to say that was a challenge. And you know what? The worst part wasn't the Nerds candies that you slurp up. It was that, the Swedish fish. Which,
oh God!
This is my tying fish back to plastics, by the way. These actually get slimy like a real fish once they've been soaking in Sprite for a long time.
They're probably made entirely of microplastics.
I feel like this is a microplastic fish right here. Yeah.
You didn't have to finish it for us, Scott. We were proud of you just for trying it. You could have stopped.
Yeah.
That's how dedicated I am as your co-host of D.O.R.K.S.
I appreciate it. And next week, whatever drink looks less good, you can assign to me. But since you're the one who picks the drinks, I will still ask that you try to pick 2 drinks that look good so that I don't suffer too much. But the next week you can pick.
Thank you. I will try to go for 2 much more appetizing drinks. So
thank you for suffering for D.O.R.K.S. I appreciate your sacrifice to our shared effort.
What can I say? I'm a martyr. Well, thank you both. This was awesome. I really enjoyed it.
Thank you for having me.
Yeah, thank you very much.
Yeah, the talks were fascinating. I really appreciate both of you putting the talks together. And if anybody has a friend who might want to watch the show but couldn't make it here today, we put the videos on YouTube, and we have a D.O.R.K.S chat Twitter account where we update about upcoming shows. And so our next show is Stephen Munger, who's going to be talking about smell and communication with a bit of a tie into COVID. And Simon, who's Dr. Oji's husband, whose last name is a hyphenated last name, but it's slipping from my memory, and he studies brain and language, and he's going to be giving a talk too. And so it should be a series of fascinating talks. And I'll have a fun story about how I asked Stephen Munger the weirdest question he's gotten in his career as part of my research for Soonish. And that's how Steve and I met. So anyway, thank you so much, guys. This was a ton of fun. And I hope folks join us for dorks in the future.
Thanks to everyone for watching and sending questions.
Yep. Bye, everyone.
Bye.